So we're going to do a couple more major body revisions, radical body plan changes in invertebrates, and then we'll jump in and start looking at examples of evo-devo within a single species. Cephalopods, which are the squid and octopus, a couple other groups in there also, including the nautilus and cuttlefish. They're the most advanced in terms of neurologically and intelligence and diversity of form of all of the mollusks. But they are mollusks, so they are part of the group that includes clams and snails and slugs and a few other things. Now, cephalopods have a dramatically different body shape and way of life than the rest of the cephalopods. Than the rest of the mollusks. The earliest mollusks looked like monoplacophorans. Now, they're still around. You probably don't know them. They're not very complex. They have a very simple shell without much structure, but it is hard. Calcium carbonate, it protected them. They have a muscular foot, and they have this structure called the mantle. Now, the mantle is a unique feature in the mollusks, and it excretes calcium carbonate, and actually is what makes their shells. And just like turtle shells, snail shells are attached. They're part of their body, so they can't pop out of their shell. And that's excreted by the mantle. Now, some of the earliest cephalopods had some modifications that were beginning to look a little bit like modern cephalopods. So they had this dramatic change in the shape of their mantle. Now, this is important. So notice, if I asked you what the anterior to posterior axis was of the monoplacophoran, it's pretty easy, right? Mouth is here, anus is down here, right at the end, so head and tail. It's easy to tell. These guys, you look at the same thing, say, well, here's their head, here's their tail, but internally, and especially if you know about the mantle and its origin, notice that they've got this kind of a bend now in their anterior to posterior axis. So instead of really being front to back, they're more like front, going up, do a U-turn to get to the back. And in squid, this has been taken to an extreme. Octopus and other cephalopods the same way. So their head and their anus, their excretory areas, actually are fairly close to one another, and they have this U-turn with the large mantle that is still there. Now, in most of the cephalopods, the mantle no longer excretes a shell. In the nautilus, it still does, and they have these very complex, quite beautiful shells. In squids, they still have an internal mantle that provides some rigid support for their body, but it's not an external shell. And then, of course, in the higher squid and the octopus, they've completely lost the shell. They still have a mantle, but it no longer excretes calcium carbonate, and they no longer have a shell. So the other major change, notice, was an elongation of this area around the mouth into these appendages, these tentacles. And distalis is the gene that turns on and grows appendages in insects and in vertebrates, and in fact, it does it in most organisms that grow appendages. So these guys use distalis, they grow appendages also. That's maybe an example of a deep homology. The same gene turns on and grows somewhat similar, although very different looking appendages. Okay, so just know that squids basically have this U-turn, this twist in their anterior to posterior axis, development of areas around the mouth into tentacles and arms, and so they really are a unique and a very, very successful group, but incredibly different from even the other mollusks. The last example of a major body plan revision we're going to talk about are the echinoderms. So echinoderms are one of two major groups within the deuterostomes. There are other minor phyla, but we basically have the two big ones are the chordates and the echinoderms. And echinoderms include starfish, sand dollars, sea cucumbers, and a few other things. Now, the remarkable thing about the echinoderms is, although they are a member of Bilateria, as adults, they do not look bilaterally symmetrical. They have this five-fold radial symmetry. So starfish, right? You can cut a starfish 5 different ways and have equivalent equal halves. Whereas with most Bilateria, you can only do that down the midline. However, obviously from their phylogenetics, from DNA, we know that they are deuterostomes. We can look at their early development and see the way that the embryo develops that puts them in the deuterostomes. So we know very clearly that they are members of the Bilateria. But even if we didn't, there'd be a good bit of evidence of data by looking at their larvae. So many of them have these bilaterally symmetrical larvae where there is a clear left hand and a right hand. So they've maintained that ancestral deuterostome, well, even more ancient than that, right? Bilaterian feature. They've maintained it, but only in the juveniles. And there's been very little work as to the actual genes that do this, but this is maybe more than even any other that we've talked about, a massive revision, a change in the basic body plan of these organisms. Okay, now we are going to do transition, and we're going to talk about changes and evolution of features within a species. One of the best studied systems for this type of a thing is the evolution of coat patterns and coat color. Now, this is a great model to study because it's very variable, so there are lots of different examples that you can look at, and it's something that changes very often and frequent, sometimes even within a species. So here we have two jaguars. We have the melanic form, sometimes called the black jaguar or black panther, and then we have the one that's more common with the spots. And so we know a lot about melanism and what causes pigmentation to occur, and so we can begin to look at a fairly small subset of candidate genes, which is important, particularly if you want to do some work or at least do some understanding of organisms like leopards that are not a good model organism, right? And so we are going to look at melanism as we investigate and begin to understand and explain variability and changes within a species. And the basic, very simplistic, but the basic question we want to ask is, do we see the same basic pattern when we look at changes within a species as we observe when we're looking at changes between a species? Now, to remind you, our paradigm is that most, not all, but most of the changes that we see between different major groups of organisms are changes, mutations, in the regulatory elements of these key developmental genes. Okay? Now, we're probably not going to be looking at key developmental genes for something as superficial as color. These are things that occur at the very, very end of development. And so although they may still technically be termed developmental genes because they're turned on and these patterns are developed, for most organisms, they're developed in utero, right? But they're really at the very, very end of development. But it's still, we're trying to answer that same basic pattern. Are we looking at changes in the genes or changes in regulation? So we're going to use melanism as an example, and then we'll draw some general conclusions after we look at the melanism example. Okay? So first thing, which genes control the diversity within a species? Now this is going to vary greatly depending on the species, right? And so this is another reason why color is so good, because color is something, and often the same genes, melanin and related pigments, are often involved in really widely distributed organisms. So everything from insects to mollusks to vertebrates use melanin as a pigmentation gene. So that's nice, we can identify this, but realize that's just for this one specific case. In other cases, we may not know what genes we need to look at. So I'm going to talk about at the end of the next, I think it's 3.4b, I will look at how we locate genes if we're not sure what genes we should look at. Okay? But for now, just remember the melanin example, and then know that we may need to do some work to evaluate the target genes that we're most interested in looking at if we want to understand phenotypic diversity within a species. Okay? And the cool thing about melanism is not only do we see interspecies diversity, but we see lots of intraspecies diversity also. So, intraspecies diversity within a species, but we also see interspecies diversity. So, for instance, we have tigers, which also have these melanic portions of their skin and fur, but in a very different pattern. By the way, I don't know if you know this, but most animals with patterns, that pattern actually goes all the way down into the skin. The melanin is produced and pigments the skin as well as the coat. So if you shaved a tiger, I think I'd be interested to look. Maybe we should Google it, right? But I think you would still see stripes on their skin as well as in their coat. So that's just a little FYI, but that's what we're looking at. And so basically what we want to know is, are these same genes involved here? Does the gene that cause stripes in tigers cause spots in the leopard, and then maybe even across other feline, and we could even look beyond feline species. Okay, so we'll answer that question. Number 3, are the same genes responsible for convergent traits? So here's a remarkable example of convergence. This is the melanic form of the jaguar, and most of them have the spots, but there are some, in even some areas it's even a little bit more common than in other areas. This is a jaguarundi. It's a small cat, a wild cat. Used to be here in South Texas. There's maybe still occasional visitors, but now their range has been pushed a little bit farther south. But they're still around. And they have a very similar look to the melanic form of the jaguar. They're only distantly related. The jaguarundi is more closely related to the small cats, whereas the jaguar is more closely related to the larger cats like lions and jaguars and things like that. By the way, there are both melanic leopard, which is these are leopards here, and they're native to Africa and the Middle East and Asia. And there's also melanic forms of the jaguar. So we could look at leopard compared to jaguar. We could look at the jaguarundi, where basically the entire species is dark, although there is some variability in how dark they are. And we can start to answer these questions. And we'll come back to these, but I'm just posing these questions for now. And then number 4, what is the mechanism for these interspecies changes? So when we look at a change from jaguar to jaguarundi or leopard to jaguarundi, are we going to see the same genes? And even if we do, maybe what we will see is the same end result, but different mutations that get us there. So if this represents turning off the gene, maybe we could do that in more than one way. And particularly if we have a complex regulatory complement that controls that gene, perhaps mutations to multiple regulatory elements or additions of new regulatory elements might actually result in convergent similar outcomes. But we could evaluate that. If we know the genes, and if we sequence and look at their regulatory regions, we could evaluate whether it's the same gene, whether it's the same change in those same genes or not. And so we're going to do that in the next discussion. We're going to expand our discussion of melanism. We're going to look at it across a wide range of organisms, particularly for one gene that's really critical in the melanin pathway.